start with the next recording has started. Okay, good. The next topic, which is extremely important and comes up multiple times. So you're welcome. So just like we will often talk about cellular injury and about getting some feedback, so be sure everyone's muted. So just like we talked about cellular injury and mechanisms of injury and apoptosis and necrosis last week, which again will come up multiple times, this material will also come up multiple times throughout the semester because we will apply those concepts both from the first class and this class to multiple body systems. So this particular lecture, these topics will talk about what happens, what is the way the body uses, what ways does the body use to protect itself, and what happens if there is tissue injury, and how that can lead to wound healing, or if in the setting of an infection, how does the body try to fight that infection? And that's the topics for today. So we're going to start with the body's first lines of defense. And the first line of defense barriers and the inflammatory process are the innate immunity. This is not something that the body doesn't have to be exposed to anything first. It's not something that's learned. This is natural, and it's the same response no matter what type of injury or what the body is exposed to. The third line of defense is the acquired immune system, and that is our more strategic, more sensitive response to a particular foreign invader, particularly pathogens like bacterial and viral infections, which is what we will focus on. So we'll begin with the first line, which is natural barriers. And part of the barrier contains mucus, which is where foreign material can be captured. The microbiome is present there, depending on where we're at, which can also assist. And if foreign material or bacteria, viruses, different types of pathogens get contained in this area, the body can help expel what's in the mucus by either coughing, vomiting, urinating, all those ways to get it out of the body. The other barrier is the epithelial cells, not to be confused with the endothelial cells, which is another group of cells we'll talk about a lot. But epithelial cells are the cells that line body surfaces. Those endothelial cells are the ones that bind or the ones that are located in the vessel. So these epithelial cells have those cell adhesion molecules which help to keep them close together. They are also adhered to the basement membrane. They will have mucus on top of them or cilia. So this is kind of a picture of them all together. So we have this is a picture of intestinal epithelial cells, that this picture of these brown lines and this yellow circles, these are all ways that the cells are attached to each other, so it's hard to breach that barrier. These villi can trap substances. There's inner and outer mucus. The inner mucus doesn't necessarily have as much of the microbiome as the outer does. And then if stuff got stuck here, certainly with diarrhea, we could get rid of some of those substances. Let me go back to the screen here. And then beyond that epithelial layer is some of the other cells that can help in our defense. But if we can keep anything from even breaching the border, then we don't have to worry about the injury itself. The second line of defense is the inflammatory process. And this process can be applied to any tissue injury, not just infection. which is obviously one of the big ones. But any injury, tissues will respond in a non-specific, rapid way. It's always the same. There's vascular and cellular phases. So the important part of this lecture is to link the symptoms or clinical manifestations of what you see when someone has inflammation to the process underneath. So what are some of the symptoms that you see when someone has inflammation that you can see visibly, like on the skin, if someone had some kind of injury? Say them or type them. Or, oh, Dawn is typing.
Erythema.
Erythema. Redness, good. Warmth. Swelling. Don't worry, Dawn, no points for spelling. Yeah, so the first things that you're saying, that erythema, warmth, and swelling, or edema, those are the symptoms of the vascular phase of the inflammatory process. The very end of the vascular phase, there will start to be some beginning of what is usually more clear drainage. The second stage is the cellular phase, and it's called cellular because different cells, non-specific cells of the immune system, get involved, and there typically is an increase in exudate at that time, which is drainage. Exudate does not have to be pus, that's just a type of exudate. So keep those symptoms in mind that you see, because you're going to have to keep linking the symptoms to what's happening in the body. So the first phase is the vascular phase, and those symptoms are the erythema, warmth, edema, and the early, more usually clear exudate. And this occurs because multiple things are happening, and most of them are happening at the same time, which is sometimes what's difficult about the inflammatory process. Is it's not where I can say this happens and then this happens and then this happens and then this happens. These are all of these things in the vascular phase are happening at once, and they involve mast cells, plasma protein systems that are mentioned there, complement coagulation and kinin, and the prostaglandins. So we will focus in particular on the mast cells and the prostaglandins. The complement cascade amplifies this response, and so does the kinin cascade. The coagulation cascade we'll talk more about next class, but this does get triggered by the inflammatory process too, because tissue injury can obviously cause varying degrees of bleeding, so we will need that taken care of too. So mast cells are cellular bags, or little bags of fluid, essentially, with some substances inside, close to blood vessels. So if I have here, let me draw a little bit here. If I have a blood vessel here, which, like we talked about last class, like a capillary would be feeding, that would be part of the extracellular matrix, and the capillary would be feeding the whatever surface is here, whatever epithelial surface. If there is an injury to this area, mast cells that live next to vessels will be activated. And their activation means that they're going to spill out substances. One of the major substances that mast cells release when they open is histamine. And when this occurs, several things happen. So histamine is responsible for erythema, warmth, and swelling, because histamine will cause, at first, a constriction of vessels, especially the artery part of the circulation, but will cause dilation of the capillary venules and those small veins, because we want to trap, we want to trap the injury in one section. So the arterial side clamps down and the venous side dilates. So everything is slowed down here. So if we have that, we have dilated vessels, that area will be warm and will be more pink in color just because of the vessel dilation. So we've already achieved two of those symptoms that we talked about. The other thing that the mast cells do is cause endothelial cell contraction. Last class, when we talked about the types of edema, we talked, which is that particular class, we focused on the types related to things that favored filtration, like an increase in hydrostatic pressure or a decrease in oncotic pressure. In this setting, we're going to have an increase in filtration of fluids because we're actually changing the vessels themselves. So if this is our vessel where those mast cells were near and we had our injury above, besides the vessel dilating and the arteries clamping, so we've kind of trapped everything in this one section, those endothelial cells that line the vessel, the histamine will actually cause them to contract so that when instead there'll be pores or spaces in between the endothelial cells, which makes it easier for proteins and water to leave the vessel. So that means histamine, when it causes contraction of endothelial cells, increases edema. So once we let the fluid out, obviously that increases swelling, but these pores could also allow some protein and platelets and other things to come out, which will also draw fluid into the interstitial space. So we should expect significant swelling. The more swelling, the worse the injury. So we got all of those vascular symptoms because of mast cell activity. And the prostaglandins and the plasma protein systems are only going to amplify that response. So the complement system helps by can cause direct destruction of foreign materials or pathogens or mark them for later destruction. And they also contribute to vasodilation. The kinin system assists with vasodilation like histamine and contributes to pain. So those systems are occurring at the same time and they're amplifying the histamine response. So the question is, how does contracting make the response, make the fluid increase? So it looks like Karis is asking, can everybody see the screen when I have the PowerPoints up? Is anyone having trouble seeing that? Oh, okay, good. Never mind. So if let me see, if an endothelial cell is here, let me pick a different slide so I can see it a little better. The endothelial cell itself actually has those actin and myosin filaments that can lead to contraction. And contraction in this, and when we talk about muscles and then when we talk about these cells, means shortening the fibers. So if these contract, the myosin pulls these in shorter, so the endothelial cell becomes smaller when it contracts. So if all the endothelial cells are contracted, there will be a space between the endothelial cells instead of being like they're supposed to be, which is resting next to each other. There is a small pore there. We could force fluid out under the setting of hydrostatic pressure, water, etc. But this is a much more efficient way to get fluid into one spot. So yes, it's very, very efficient. So the thing about the plasma protein systems that are the most important, we will talk in much more depth about the clotting system. But just to say that the platelets are going to come out and be a part of this process as well as the clotting cascade. We'll just get into more details about that next class. But the main issues, the main things that the complement and kinin system do are to amp up the inflammatory process by increasing vessel dilation and contributing to pain response. The other part of the vascular process, or the vascular side of the inflammatory process, is prostaglandins. And these are created by injured cells and their interaction with particular enzymes. So here's our basement membrane, and here's our epithelial cells that look like, that's a terrible drawing. Okay, those are our epithelial cells, and they're on a basement membrane, and then there's this vessel here with mast cells, and there's an injury that occurs, and the stuff that we talked about is already happening. The complement cascade, the coagulation cascade, the kinin cascade, the mast cells are releasing histamine. There's the expected erythema, warmth, and edema. So, and it's a much more significant edema in a specific area. These cells that were injured, their cell membrane is made of phospholipids, because that's what the cell membrane is made of. When they are injured, a specific amino acid that is part of the cell membrane is released, and that's arachidonic acid. In the bloodstream, these enzymes, cyclooxygenase enzymes, or COX-1 and COX-2, are always present. If they are allowed to interact with arachidonic acid, they will produce prostaglandins. The main prostaglandins are created by COX-2 because those are the pro-inflammatory prostaglandins that increase inflammation by increasing vessel dilation, increasing the pain response, and also sending a message to increase fever. So you have to remember, does anyone remember how the body regulates temperature? Cara says, Michelle. It is through the hypothalamus. So the hypothalamus is our thermostat, essentially. It has a set point that it's set at, and that is, in general, average. That's an average normal temperature. It is possible to have a normal temperature within a range between 97 and, in the 99. Some people's normal temperature varies. If the hypothalamus is sent a message, which will happen during inflammation and especially during infection. Prostaglandins can increase the temperature set point, but others of the cytokines and interleukins can increase the set point much more efficiently. But if these prostaglandins that were created by the interaction of arachidonic acid and COX-2, if they go into the hypothalamus and increase the set point to 100 degrees Fahrenheit, then the body has to do that. So just raising the temperature set point doesn't increase the person's body temperature, but it does cause a chain of reaction to do things in the body to increase the temperature itself. This is a adaptive response. The cells of the inflammatory process, as well as of the adaptive immune response, work better when the temperature is a little higher, and so it is adaptive to increase the temperature slightly. So temperature goes up, the way the body can increase the actual core temperature is by muscle contraction. So the person feels the chills. What it is the contraction of their muscles, and will have the feeling that they need to add blankets to themselves. So they will increase their heat. The body heat will increase. So the experience of the chills is actually the body's attempt to raise the temperature up to the new set point. So potentially a fever could be a part of this early inflammatory response. Usually that is much more profound once we get to the part of the immune system that is supposed to deal with infection. So there's our vascular phase. Now we're getting into the cellular phase. At the end of the vascular phase, because of the degree of fluid and different proteins from the vessel in the confined space, sometimes people will see some weeping of starting to see some fluid. But there will definitely be an increase in the amount of exudate during the cellular phase, because this is when we get more of the non-specific cells of the innate immune system to the area, which is why it's called the cellular phase. And there will be an increase in exudate, and by getting more of these cells, we could begin either healing or we could connect to the adaptive third line of defense, which is acquired immunity. So, in the cellular phase, we know that the endothelial cells, not only did they contract in response to histamine to allow fluid and proteins to leave, they also produce specific cell adhesion molecules that pull white blood cells to the area of injury. Those adhesion molecules are called integrin and selectin. The first ones we're going to see, which is the first part or the first thing that helps determine our exudate, is the neutrophils. Those are the first of the cellular response. So these, we have our vessel, we have those endothelial cells that have contracted. They develop or push out these specific proteins that will help pull white blood cells to their surface. In this case, we want to pull neutrophils. This grabbing of the neutrophils will assist them in leaving the vessel and going into the tissue to the area of injury. So we had to pull them in. They are the first line of the cellular response, and their sole job is to remove debris. So if it's just a wound from, a burn that's not infected, or maybe you fell, it's traumatic, direct injury, whatever the original source was, we have to clean up the dead tissue. So one of the jobs of the neutrophil is debridement of a wound. So it's going to get rid of debris, and it also, inside of it, has a particular enzyme that helps with to mediate tissue destruction, because we want to get rid of some of the bad tissue, but we don't want to make it so that we harm healthy tissue. So the neutrophils get there, they engulf any foreign material. That foreign material or microorganisms will bind to the lysosomes inside the cell, which we learned were the organelles that contain the enzymes that can damage whatever they bring in. And those lysosomes can eat up the little pieces. And then they don't divide, they will spill their contents. And that is how we increase the amount of exudate. So we have these neutrophils that have lysosomes in them. They're going to take in foreign material, which could be pieces of cellular debris or could be microorganisms, and they're going to interact with those and cut them up into little pieces. And then instead of dividing, they're going to open up and spill their contents into the interstitial space. And this will provide the body with two, yes, it does look like a Pac-Man. It's my beautiful drawings. So when it spills its contents, it helps with two things. Remember, there's lymph channels here. Some of that fluid, they broke it up into little pieces, will then be filtered and taken away by the lymph vessels and filtered out of the body into the lymph system, which eventually drains into veins and goes away. So that helps clean it up. The other thing it's doing is when it opens up, there's also lysosomes that are released, which can help with debridement. And then we have that protective alpha antitrypsin so that we don't harm stuff, the other tissue around us that hasn't been damaged. So the action of the neutrophils is margination, and that's the endothelial cells sort of capturing the neutrophils out of the general circulation and holding them to the vessel wall. And then transmigration, which is the neutrophil leaving through that pore into the area of injury. Chemotaxis is them traveling towards the area that's the problem, and phagocytosis is the Pac-Man thing, is them getting rid of the foreign material, in this case, infection. The exudate will increase when this occurs, and that helps us get some information from our assessment too. So the exudate is helping with wound debridement and will help filter and clean up the wound, but we can also get some information on exam by looking at the exudate, because it'll be a mixture of the fluid and cells that are present in this injury. So we would expect serous exudate early. If it's purulent, that means that when the neutrophils dumped their contents, there was infection in there. There is bacteria that's been broken down, and we're smelling or seeing that reaction. If it's hemorrhagic, that means there's more blood. There had been bleeding, more bleeding with this injury. Sometimes it's a mix of purulent and hemorrhagic. And over time, as this exudate will become more fibrinous with clot and other thicker tissue. The monocytes are the second line of innate immune cells to arrive, and they will survive and divide. So the neutrophils, oftentimes people use military references when talking about the innate immune system. The neutrophils are sort of like the first line troops, kind of like the pawns in a chess game that are there. They kind of give their lives to the cause. The monocytes, which are activated to macrophages, come later, and they are there to do a more selective cleanup because they will survive and divide. They have special recognition receptors called toll-like receptors that helps them realize when cells belong to our body versus cells that are foreign. They release interleukin, which is a very important type of cytokine, which will call other types of white blood cells to the area and contributes greatly to the fever response. It will help, like I said, call more immune cells to the area. They also release something called tumor necrosis factor, which with the interleukin increases fever and tells the liver to produce more proteins, because everything that we use in this process, the monocytes, the things we need to heal the area, the proteins that make up the cytokines and the chemokines, those are all made of protein. So we have to tell the liver to get to work. Those are the types of interleukin. And the natural killer cells are the lymphocytes that are present, are part of the innate immune system, and they're the last ones to really arrive to this area. They also use toll-like receptors to recognize cells that are self or non-self. So at the end of the inflammatory response, we are set up for either wound healing and a calming of this response and a healing of this area, or we are set up to hand off the job to the acquired immune system. So if we look at this picture, this is an early wound, and you can see there's the exudate. That would be serous exudate. This looks like an abrasion, so there is some bleeding. There's definitely some cell injury from the direct, fall. So we're at least at the stage, looking at this wound, of the end of the vascular stage, where there's some early exudate, there's erythema, there's mild edema, looks like there's been bleeding. So when we look at a wound, we can say, okay, this had to have happened because the tissue injury led to the degranulation of mast cells that releases histamine, that causes constriction of the arterial side to help with contain any bleeding, and dilation of the venous side to help increase, kind of trap whatever the injury is. This also increases redness and warmth to the wound. The histamine's other action is to contract the endothelial cells to allow fluid to escape the vessel, as well as proteins and other things like platelets that will contribute to the wound healing process, and that will increase the fluid that's drawn to this area because of the pull of oncotic pressure. And then the next response will be for the cellular side to start, will be to pull neutrophils into this spot. The other thing that's going on at the same time is activation of the protein cascades, which is the clotting cascade, which we'll talk about, which decreases bleeding. The complement and kinin cascade increase dilation of vessels and the pain response. And then the actual injury from a cell with the release of arachidonic amino acid from the cell membrane will help produce prostaglandins when it interacts with the COX-2 enzymes. So, that's a lot, right? That is the entire inflammatory process in a nutshell, and that is going to come up over and over and over again because, one, well, obviously it's our week's material, but every time there's an injury someplace, whatever kind of injury that is, all those injuries we talked about the first class, we are going to trigger the inflammatory response. So we're going to have to superimpose or put that inflammatory response onto whatever system we're talking about. Because if we can see it on the skin, but those same things happen inside the body too that will produce symptoms. It looked like somebody was typing something unless they changed their mind. So it's very, very important to keep, listening to this, to read it over. There's a couple of videos that describe it, to be sure that you can verbalize the steps of the inflammatory response from vascular to cellular, because that is going to come up many, many times. So the next part is wound healing, which we can start with. I don't say that. Okay. So phase 1 of wound healing was what we were just talking about. We needed the vascular phase and the cellular phase of inflammation to get to this time. We needed the area of injury to be infiltrated with platelets, neutrophils, and macrophages. We needed these cells not just because platelets help stop bleeding, but they also release growth factors that help form new vessels. So the only way we're going to get new vessels is from the growth factor released from platelets. So then we can revascularize the area that was injured. Neutrophils, we talked about, is a wound debrider because when it spills its contents, that means lysosomes are also released. So that helps debride the wound as well as protect areas that aren't injured. The macrophages, which keep in mind, monocytes and macrophages are not different things. Monocytes, when they become activated, are macrophages. Macrophages help further clean up debris and the mess of exudate that's been left by the neutrophils, besides the stuff that's been filtered out from the lymph system to help drain away that extra fluid. But eventually then we have to calm down the permeability because we don't want fluid to continue to come into this wound that we're trying to heal. And so we want to reverse that permeability and kind of we want to grow new vessels where we need to, but we don't need the vessels to be as dilated. So we have to calm down inflammation a little bit. So if the inflammatory response is still active, or if this area is infected, we can't really move on to phase 2 wound healing because we haven't taken care of this. So one of the reasons why wound healing gets delayed is if there's too much fluid in a wound, then we can't get this to work. If there's still an active infection going on, that's another reason why wound healing would be delayed. When we get to the second phase, which is kind of after the initial inflammatory response and then continues on for the next 2 weeks or so, we have to grow in the area where the tissue injury occurred. So oftentimes people talk about wound healing in terms of skin injuries, but this has to happen inside as well. You know, if there's an internal injury, the same process takes place. And in that, if you can recall from our first class, in the extracellular matrix, there is pieces of collagen and fibrin that are available to proliferate if needed in order to help heal in a wound. So they're there, the fibroblasts and collagen, and can be induced to proliferate, grow more, so we can have some supportive structures to our wound. The macrophages release epithelial growth factor, which induces epithelial cells to grow, to further divide, to fill in the open area. And then we can get to this point. So what I was just describing is we have this wound and we have our blood vessel in the extracellular matrix, and there's those mast cells and neutrophils and macrophages and those parts of the inflammatory response. But in this area, if we make it so that this is actually injured, in this area in the extracellular matrix is fibrin and collagen, you know, however we want to draw that can be induced to grow. And when it does, it can help scaffold the wound. And the platelets release growth factor, their own vessel growth factor, to be sure that this new area is supported with blood flow. And the macrophages release epithelial growth factor, which helps grow in more cells to this area. So that's what we're trying to do. We're trying to fill it in. The fibrin and the fibroblasts and the fibromyoblasts, which are similar because in that they have actin and myosin so they can contract, are there to help pull the wound together. So it's there to help support the growth, tell the blood vessels and the epithelial cells where to grow in, but it's also there to support and scaffold those cells and then to pull them in closer together, which is what the next phase will require. So we had our reconstruction, we filled in the tissue space with granulation tissue. We increased epithelialization to increase the cells to grow into this space. And then we have to continue to remodel and strengthen this wound. And this can take a really long time. And that's why scar tissue is constantly remodeling. So oftentimes a scar could look very, very sort of ugly, red, more exuberant in appearance. And people will think, oh, I don't like this, I want it revised. But you really shouldn't do anything with the scar for at least 2 years, because the scar will continue to soften and remodel. And the way it does that, that's not the best picture in the world, but the way it does it, the way I was taught to think about it, is that fibrin and collagen scaffold is sort of like wet spaghetti. You know, it's there, it's nice, but it's not as strong as you would like it to be. It's kind of soft and boggy if you would push on it. It could open up with little trauma, so we need that to get stronger. We need the edges of the wound to be pulled together better. We need less water in there. So when those myofibrin pieces, or the pieces that actually have the ability to contract, in a sense, they're squeezing the water out of the spaghetti. They're pulling the edges of the wound together tighter and trying to get, like squeezing a rag, trying to be sure the extra fluid goes away. And then those myoblasts or myofibers will relax. And some time goes on, and then that contraction occurs again, where we're again squeezing the rag, we're getting more fluid out, and we're bringing the edges together. So over time, the scar should be stronger and usually less visible. So that's the phases of wound healing. Obviously, there's dysfunctional wound healing, which I'm sure many of you have come across. And it could be if there isn't enough blood flow to the area. Wound healing and the inflammatory process itself is a vascular process. If we're going to induce cellular proliferation, if we're going to make cells grow, then we need oxygen to do that. So if there's any ischemia to the wound and there's less oxygen to the area, it'll be harder to do that. In fact, most people, if the wound is big enough, will actually have an increased oxygen requirement, and that might actually increase their respiratory rate. If there's excess bleeding or infection or lots of fluid in that wound, it'll delay the wound coming together. But the opposite could be true as well. If there's not enough fluid in the area and the wound gets dry, it will also cause dysfunctional healing. If the person is malnourished, there's not enough proteins in the blood in order to facilitate this growth. Some people are on medications that can delay the immune response. Some people are heavy scar formers and will have very fibrous scars or overproduction of scar tissue. So dysfunction during reconstruction is when you're trying to get that collagen together and sort of bringing it in and then relaxing it and bringing it in and then relaxing it. Sometimes it doesn't work because they don't have enough protein, so they can't make the collagen. But sometimes the exuberant response, and this can be genetic, is that they're making too much of it. So you can have two types of dysfunctional scarring that is an overgrowth of scar. One is called a hypertrophic scar, and this means that the excess collagen is there, the scar is raised, but it doesn't grow beyond the injury itself. A keloid scar also is a raised scar, but grows beyond the injury into different directions. So these are keloids because they're growing outside of the actual scar wound and have an abnormal, irregular appearance, whereas a hypertrophic scar would just be very raised. Sometimes people can't get on. These are some incidences that can decrease the amount of epithelialization. And then some people have impaired contraction. They might actually have too much tension on the wound and end up with scar deformities. And then, of course, a wound could be disrupted as well and give way. So the main thing to get from here is that if there is no infection, the end stage of the inflammatory response would be to calm down the vessel permeability and dilation, and after debridement and cleanup of the wound would be to increase fibrin, collagen, and epithelial cell growth, and then increase myofibroblasts or myofibrin in order to contract the wound as it gets reconstructed. And that it does take at least 2 years for full scar healing to occur. So at this point, it is 7:49. We'll take a little break till 8 o'clock. Let's see, where am I? When we come back, I have a couple pictures of wounds that we can look at, and then we'll move on to types of infections because I want to differentiate especially between bacteria infections and viral infections because that does predict a little bit the acquired immune response. So